Right, so let's try to answer this question for biology paper 2. So the first question is saying figure 11.1 shows two liver cells as seen under a light microscope. So we have the cell membrane and also the nucleus. So we need to know the functions of the nucleus and the cell membrane. So let me start with the cell membrane. So the cell membrane selects what goes in and out of the cell, like water and other gases. As a result, this one is referred to the selectively semi-permeable membrane. So the other name for the cell membrane is just the selectively semi-permeable membrane because it selects what goes in and out of the cell. Then when it comes to the nucleus, this nucleus stores genetic information in form of the chromosomes. So we can conclude that the chromosomes are found in the nucleus. Then apart from that one, it also has the function of controlling all the cell's activities. So the nucleus controls all the cell's activities. Now, the first question, which is question A, Roman numeral 1 is saying, liver cells produce bile. Describe the role of bile in the digestion of fats. So you need to know what constitutes bile. So we have what we call sodium hydrogen carbonate, which is the first one. So apart from sodium hydrogen carbonate, we also have what we call the bile salt or bile salts. So we can say the bile salt or bile salts. Then the last one, we have what we call the bile pigment, which is the last one. Now, usually when it comes to the bile pigment, this one, it has no use in the digestion of the fats. So you are going to find that this bile pigment, it only adds color to the feces. So it has no use when it comes to the digestion of the fats. So meaning we are going to consider two things here. The first one is going to be sodium hydrogen carbonate and also the bile salts. So we need to explain the functions of these two. So now we have the first one, which is sodium hydrogen carbonate. So as for sodium hydrogen carbonate, this one sets an optimum pH of the enzymes in the duodenum. So usually when food is coming from the stomach, we know that in the stomach there is hydrochloric acid, meaning the food is going to have the acidic pH. Now from the stomach it goes to the duodenum. So now why are we saying this one, sodium hydrogen carbonate, sets the optimum pH for the action of enzymes? You are going to find that once bile has been produced by the liver cells, it goes to the duodenum. Now, in the duodenum there, we have the pH of alkalinity, meaning it is not acidic, but it's alkalinity. So when the food is coming from the stomach, it is going to have the acidic pH because in the stomach there is hydrochloric acid. So once bile has been produced by the liver cells, we know that in the bile we have sodium hydrogen carbonate. This one is going to neutralize so that the pH for the action of the enzymes that work under alkalinity can be set up. Okay, so to set up the pH for the action of the enzymes like the lipase, that's the answer. Then when it comes to the bile salts, these bile salts, they emulsify fats. When you say emulsify fats, by definition you can say emulsification, just the breaking down of large droplets of fats into smaller droplets. That's the definition for emulsification. So you're going to find that these bile salts or bile salt, it just emulsify, thereby breaking down large droplets of fats into smaller droplets. Now once they are broken down, this increases the surface area for the action of what? Enzymes. So we can say the answer is going to be the bile salts emulsify the fats, thereby increasing the surface area for the action of enzymes. So that is the answer. Then as for Roman number 2, it is saying, state two other functions of liver cells apart from bile production. So you are going to find that when the body is having excess blood sugar, so when I talk about excess blood sugar, I'm talking about excess glucose. So the other name for the glucose is blood sugar. So once you are having excess blood sugar in your body, you are going to find that the pancreas is going to secrete the insulin. Now that insulin is going to convert the excess blood sugar into glycogen, and that glycogen is going to be stored into what? Into the liver cells. So we can say conversion of excess blood sugar into glycogen. Then apart from that one, or if you want, you can just say to store glycogen. That's a function. Then apart from that one, you can also say the liver cells, they destroy used up hormones. So once hormones are used up by the target organs, they go direct to the liver and being destroyed by the liver. So in addition, you can also say production of blood cells. So the liver produces what? The blood cells. Its production, detoxification of poisonous, like that. So you can choose. The next question. So the next one is saying, under a normal power of microscope, the cell membranes of the liver cells can be seen to be folded. Suggest why this is important for the functioning of the liver cells. So we need to explain this one. Alright, so we have 2 marks here. So the first one, you can say, when the liver cells are folded, this one is going to enhance the absorption of the nutrients and the hormones. So once the liver cells are folded, this one is going to enhance the absorption of nutrients, hormones into the bloodstream. Then apart from that one, it also increases the cell-cell interaction. Okay, so it increases cell-cell interaction. Once they are folded, cells are going to be brought together, meaning you're going to find that there will be the interaction there of the cells. Then apart from that one, it enables now, when the cell membrane of the liver are folded, this one enables the liver cells to perform the critical functions like the metabolism and detoxification. Then apart from that one, it also enhances the bile production. So those are the answers for question B. Then when it comes to C is saying red blood cells and plant root hair cells also have structural features that are important for their function. Roman number 1. State one function of root hair cells. So the function of root hair cells is just to absorb water and mineral salts. So we can say absorption of water and mineral salts. Then apart from the absorption of water and mineral salts, we can talk of anchoring the plant to the ground. So the root hair cells, they also anchor the plant to the ground. Then if you don't want to say that one, you can also say the root hair cells, they act as the storage organ. So they act as the storage organ. Then as for Roman number 2, it is saying, state the function of a red blood cell and give 2 ways in which its structure is related to its function. So the function of the red blood cell, it is to transport oxygen around the body. Then apart from transporting oxygen around the body, if you want, you can say it also transports what? Antigens on the cell membrane. So the antigens, they are found on the cell membrane of the red blood cells. So you can include that one if you want. Then how the structure is related to its function. So the first one, you can talk of the biconcave in shape. So the red blood cells, they look something like this. And then at the center here, they have a biconcave in shape, meaning here there is a space like a plate, like the way the plate is. It's biconcave. So on this part here, that is where we are going to have the biconcave shape. Now this biconcave in shape creates the large surface area for the carrying of hemoglobin. So the hemoglobin, it is carried on the biconcave in shape. So now the function of the hemoglobin now, it is to attract oxygen. Okay. Then apart from that one, we can also say the presence of hemoglobin. Now this presence of hemoglobin, we can say, which has high affinity to attract oxygen. So the presence of hemoglobin, which has high affinity to attract oxygen. So those are the answers. Try to look at the next question. So the next question is coming from reproduction in humans. So it is saying figure 2.1 shows a fetus in the uterus. So we have part X. So this part X is called the umbilical cord. Some they say umbilical cord, or you can say umbilical cord. So the function of the umbilical cord, this one is just the passage now, where the exchange and nutrients takes place. Then when it comes to the uterus walls, or the uterus wall, that is when now the implantation occurs. Then we have part Y. So part Y is called the amnion. So the function of the amnion, this one is more like a bag. So the amnion is just more like a bag where now the fetus or the fetus is found. Okay, so we have also the last one, which is Z. So this Z here is called the amniotic fluid. The function of the amniotic fluid. So the function of the amniotic fluid, this one regulates what? Temperature. Apart from regulating temperature, it also prevents the mechanical shock to the baby. Okay, so let's try to answer the questions. As for the first question, which is question A, saying, name the structures labeled X and Y. So we can say X is going to be the umbilical cord or umbilical cord. And then as for Y, it is going to be the amnion. Okay. Let's see the next question. So B is saying, describe the function of the fluid at Z. So fluid Z is called the amniotic fluid. So we need to give them the function of the amniotic fluid. So the first one, we are going to say it regulates the temperature. Apart from regulating the temperature, it also prevents the mechanical shock to the baby. Or we can say the mechanical damage to the baby. Then apart from that one, it also allows the development of the lungs. Now, why are we including this one? When you say development of the lungs or lung development, you are going to find that in the amniotic fluid, the baby or the fetus is going to be able to, the lungs of the fetus are going to be able to expand. So once they are expanding, that one is going to allow the development now of the what? The lungs. Then apart from that one, you can also include this one, which we are going to say digestion development. So you can say digestion development. And then apart from digestion development, you can also include the last one, which we are going to say skin development. Okay. Then as for C, it is saying suggest 3 ways in which the composition of the fetus blood changes as it passes through the placenta. So question C leads. Suggest 3 ways in which the composition of the fetus blood changes as it passes through the placenta. Okay, so to answer this question, let me just bring you back to the diagram. So we have the umbilical cord, which is the passage where now the nutrients and oxygen and other gases passes. So we are going to find that when the blood is coming from the mother, let's imagine it is coming from the mother. The blood is coming from the mother. Let's imagine it is coming from the mother like this, going to the fetus. So if the blood is carrying oxygen, that oxygen is going to be used up by the baby. Then the baby is going to give out now carbon dioxide. Similarly, the carbon dioxide now is going to go now to the what? To the mother. Apart from that one, also the nutrients present in the blood, they are going to be absorbed by the baby. So the baby is going to take the nutrients. Then the waste metabolism, they are going to be taken out from the baby through the umbilical cord into the placenta and goes to the excretory organs. Now, to make you understand what they're talking about here, we can talk of oxygenation because of what? So we can say the first one just because of oxygenation. Okay, that's the first one. Then number 2, we can also include, which we are going to say because of the nutrients intake, the nutrients of intake. Then apart from the nutrients intake, because of the removal of waste metabolism or waste removal of metabolism. So those are the answers. Okay, so the next question please. Figure 11.1 shows some of the regions of the alimentary canal in a human. So we have part A. So this part A is called the esophagus. You can say esophagus or gullet if you want. Then apart from A, we also have B, which is called the stomach. Then apart from the stomach, we also have C, which is the liver. So you can say the liver. Now, as for the first question, which is question A, saying, name the structures labeled A, B, and C. So we are going to say A is going to be the esophagus or gullet. So the function of the esophagus or gullet, this one is just the passage where now the food passes when it is coming from the mouth. Then as for B, we can say B is the stomach, an elastic bag which stores food or the nutrients temporarily. Then as for C, C is going to be the liver. It has many metabolic functions like bowel production, heat production, destroying of used up hormones, and so on. And in addition, we also have this structure which is called the pancreas. We also have the small intestine and also the pancreatic duct. So let's see the next question. So the next question is saying, state one function of the pancreas. So this pancreas here, we need to state its function. So we can say the first function of the pancreas is going to be, we can say it produces the hormones. Now these hormones include the insulin and the glucagon, so production of the hormone. Then we can say insulin or glucagon. Then apart from the production of the hormone or hormones, it also secretes what we call the pancreatic juice. Okay, so it secretes pancreatic juice. So the pancreas has two functions. So those are the functions. So you can just choose one because here we are having one mark. Which is question C. It is saying, in people with cystic fibrosis, the pancreatic duct may become blocked. Suggest and explain what the effect of this would be. So let's see the pancreatic duct. So at this point, that is where now we are having the pancreatic duct. Now, closely here. Near the pancreatic duct, at this point, we are going to have the duodenum. Okay? So we are going to find that since the pancreas is producing the pancreatic juice and also the hormones, meaning if this one is blocked, the duodenum is not going to receive the pancreatic juice. Apart from that one, the liver, it has the function of the bowel production. So as you can see, this tube here, it also passes through the pancreatic duct. Meaning if the pancreatic duct is blocked, the bowel won't reach the duodenum. So those are the effects. So we are going to say the first one. When the pancreatic duct is blocked, the bowel won't reach the what? The duodenum. Then apart from that one, when the pancreatic duct is blocked, the pancreatic juice and also the hormones like the insulin and the glucagon won't reach the target organ or the target organs, and of the target organs, meaning the insulin and the glucagon, they won't reach the target organs. The next question, which is D, Roman number 1, it is saying, with reference to the alimentary canal, define the term absorption. So we can say absorption is the process by which digestive nutrients are taken into the bloodstream. we can just say the process by which digestive nutrients or digestive foods are taken into the bloodstream. That is the absorption. Okay, then as for Roman number 2, it is saying, name the parts of the alimentary canal in which there is the most absorption of, we have the sugars and also water. So when they talk of sugar, they're just talking about glucose. So the part where the absorption of sugars mostly take place, it is in the small intestine. Now, why are we saying in the small intestine? Because the walls of the small intestine, they have what we call the microvillus. So these microvillus, they look something like this. They have the central part, which we are going to call this one as the lacteal. And also, they have the blood capillaries. So you can just draw it roughly like this. They have also the blood capillaries. Now, you are going to find that specifically where the absorption of the sugars and the amino acids takes place, they take place in the blood capillaries. So in the blood capillaries now, that is where now the absorption of sugars and amino acids takes place. Then when we talk of the central part of the microvillus, which we are calling it as the lacteal, that is where now the absorption of fatty acids and glycerol takes place. So we can say as for sugars, it is in the small intestine, specifically in the microvillus. It comes to water. So the absorption of water, most of the absorption of water takes place where? In the large intestine. So in the large intestine, that is where now the absorption or most of the absorption of water takes place. Then as for Roman number 3, it is saying explain how assimilation differs from absorption. So when you talk of assimilation, this one is just the usage of the digestive food into the alimentary canal. Okay, so the usage and incorporation of the digestive food into the alimentary canal. While when you talk of absorption, this one is just the intake of digestive food into the bloodstream. That's a difference. Let's see the next question. So the next question is coming from sense organs. And it is saying, the diagram below shows the structure of the human eye. So we have part A. So part A, this one is called the iris. So part A is called the iris. Then as for part B, this one is called the choroid. Then as for part C, this one is called the sclera. So it is called the sclera, something like this. Then as for D, so D is called the retina. So D is called the retina. Then as for E, this E is called the optic nerve. Okay, so there are a few parts which are remaining on this part. We also have this part here, this one here, which we are going to call it as the fovea centralis, something like this. Okay, then apart from that one, we also have this space here. This space here, it is called the pupil. Then you also have this one here, it is called the lens. Then the last part, let me just include the last one. This part here, it is called the cornea. There is a difference between the cornea and the sclera. So the sclera is the white part of the eye. Then when it comes to the cornea, this one, it is just the transparent part of an eye. So meaning, on the cornea, that is where now the diffraction of light takes place. Okay. We also have the liquid which is found in the eye. It is called the vitreous humor. Now, as for the first question, it is saying identify part B and C. So as for part B is called the choroid, the black part. The black part is called the choroid. Then when it comes to part C, so part C is called the sclera. Let's see the next question. Explain the effect on a person's vision if part E is cut. E, it is called the optic nerve. So now the optic nerve sends the impulse or the visual impulse to the brain. So once part E, which is the optic nerve, is cut, you're going to find that it is going to fail to send the visual impulse to the brain. As the result, the person is not going to see. You can say, cutting part E, resulting the eye failing to send the visual impulse to the brain. That's the answer. Then apart from that one, we can also say the person is not going to see because part E sends the visual impulse to the brain. Alright, so the next question, it is saying part D can be damaged by very bright light. So we have part D. So part D is called what? The retina. So the retina contains two cells or two sensitive cells. We have what we call the cones. And also the last one, which we are going to call it as the rods. So the retina consists of two sensitive cells, the cones and also the rods. So now usually the cones, these they are used for the identification of the what? The colors in the bright light. Then as for the rods, these are used when you are in the room where there is low light intensity. So those are the uses. And rods are able to form blurred image, meaning you're not going to see the color. So for cones, they specifically identify the color. Okay. So now the question is saying we need to describe how part A helps to prevent part D in very bright light. 4 marks. I'm identifying that part A is the iris. So now how does the iris helps the damaging of part D, which is the retina? So now to answer this question, you're going to find that this space here is called what? The pupil. So when there is very bright light, you are going to find that the iris muscles, they are going to contract. Specifically, the circular muscles, they are going to contract. Then the radial muscles, they are going to relax. Now when it comes to the iris, it has two muscles. So let me do this. It has what we call the circular muscles, which I'm drawing here. So these are called the circular muscles. Suppose we have two circular muscles here. Then we also have what we call the radial muscles, which are like this. So this one is the iris. Now the space here that you can see here, it is called the pupil. Okay, that's the pupil. Now you are going to find that when there is very bright light, the circular muscles, these, they are going to do what? They are going to contract. And then the radial muscles, they are going to relax, thereby preventing high light intensity to reach the retina, because these sensitive cells, like the cones and the rods, they are destroyed by high light intensity. So they are going to be destroyed by high light intensity. So to prevent part D from being damaged, when there is very bright light, the circular muscles, they are going to contract, and the radial muscles or longitudinal muscles, they are going to relax. That's the first one. Once they contract, and once the circular muscles contract and longitudinal muscles relax, you are going to find that the pupil is going to be reduced in size. Okay? Meaning when there is very bright light, the pupil reduces in size due to the contraction of the circular muscles and relaxation of radial muscles or longitudinal muscles. So that is the answer. So that was the last one. Thanks for watching and see you next time. Remember to subscribe and share this video to your friends.